Friends, welcome to Cultura channel. I am Dr. Varsha Gaitonde and this video is about the syllabus of genetics and plant breeding for undergraduate students as proposed by the 5th Dean's Committee, and this is for different semesters. Let's begin. There are 7 courses where the subject of genetics and plant breeding is divided into. The first part consists of GPB 121, that is fundamentals of genetics, which has given credit of 3, that is 2 plus 1. Here 2 indicates the theory classes and 1 practical class. In part 2 means in semester 3, 4 and the other course is also involved in semester 4 only. In semester 3, GPB 211, that is fundamentals of plant breeding, is again a 3 credit course. 1, that is 2 plus 1. Then in 4th semester, GPB 211, that is principles of seed technology, again 3 credit course, and GPB 222, that is commercial plant breeding, which is a 1 plus 2 course, means 1 is theory and 2 practical classes. In part 3, that is 5th semester, GPB 311, that is crop improvement for kharif crops, that is crop improvement part 1, which is a 1 plus 1 course. In 6th semester, GPB 321, that is crop improvement part 2, that is crop improvement for rabi crops, again 1 plus 1 course. GPB 322 is micropropagation technologies, which is a 1 plus 2 course, that is total 3 courses are allotted for this particular course. So let us see the detailed syllabus for individual course. First course is fundamentals of genetics, GPB 121. Here as a theory portion, topic covered are pre and post Mendelian concepts of heredity, Mendelian principles of heredity, cell division for mitosis, then cell division in meiosis, probability and chi-square test. This is also related to Mendelian genetics. Then dominance relationships and gene interactions, epistatic gene interaction with examples of complementary, supplementary, duplicate gene interaction, epistatic gene interaction with examples of masking, inhibitory, polymeric and additive gene interactions, then pleiotropism, pseudo-alleles, multiple alleles and blood group genetics, then sex determination, sex-limited, sex-influenced and sex-linked traits, sex linkage, linkage and its estimation, crossing over, where the introduction and mechanisms of crossing over are discussed, chromosome mapping, structural changes in chromosome, numerical changes in chromosome, mutation introduction, then characteristics and classification, mutagenic agents like physical, chemical mutagens, then induction of mutation, methods of inducing mutations and CLB technique, qualitative and quantitative traits, especially polygenes and continuous variation, multiple factor hypothesis, cytoplasmic inheritance, genetic disorders, nature, structure and types of genetic material, proof for DNA as a genetic material, replication of genetic material, genetic code and protein synthesis, transcription, mechanisms of genetic material, then translation and mechanisms, gene concept, it includes gene structure and function, gene regulation, it includes the operon concept, lac operon, tryptophan operon, positive regulation and negative regulation. Then practical topics of this particular course, microscopic studies, its parts and types, then study of cell structure, experiments on monohybrid test cross and back cross, experiments on dihybrid test cross and back cross, experiments on trihybrid test cross and back cross, Then experiments on epistatic interactions, including test cross and back cross. Experiments on epistatic interactions, including test cross and back cross. Then strains and their preparation. This is all related to cytogenetics. Fixatives and their preparation. Practices on mitotic cell division. Practices on meiotic cell division. Experiments on probability. Then experiments on chi-square test. Determination of linkage and crossover analysis through 2-point test cross and 3-point test cross data. Study on sex-linked inheritance in Drosophila. Then study of model on DNA and RNA structures. Some of the references are also coined here. Let us move to the second course, that is GPB 211 Fundamentals of Plant Breeding. The theory part includes plant breeding, its concept, nature, objectives and role, historical developments of plant breeding, major achievements and future prospects, genetics in relation to plant breeding, modes of reproduction, pollination, apomixis, self-incompatibility, then male sterility and its genetic consequences, domestication, acclimatization, introduction, centers of origin or centers of diversity, component of genetic variation, heritability and genetic advance, genetic basis of self-pollinated crops and pure line theory, breeding methods in self and cross-pollinated crops, mass and pure line selection, hybridization techniques, handling segregation populations like pedigree, bulk, backcross methods, multi-line concept, concepts of population genetics which includes Hardy-Weinberg's law, genetic basis and methods of breeding cross-pollinated crops, population improvement and modes of selection, heterosis and inbreeding depression, development of inbred lines and hybrids, composites and synthetic varieties, breeding methods in asexually propagated crops, clonal selection and hybridization, wide hybridization, pre-breeding, polyploidy in relation to breeding, mutation breeding, its methods and uses, breeding for important biotic and abiotic stress, breeding for important abiotic stress, then biotechnological tools like DNA markers, introduction to markers, then marker assisted selection, participatory plant breeding, intellectual property rights and patenting, that is IPR, then plant breeders and farmers' rights. Let us move to the practical course of plant breeding. Plant breeders' kits means what all a breeder has to carry when he goes to a field, then study of germplasm of various crops, study of floral structure of self-pollinated crops, then floral structure of cross-pollinated crops, emasculation and hybridization techniques in self-pollinated crops, then the same in cross-pollinated crops, consequences of inbreeding on genetic structure of resulting population, study of male sterility systems, handling segregating population, methods of calculating mean, range, variance, standard deviation and heritability, then designs or experimental designs used in breeding, analysis of randomized block design and components of genetic variance, to work out with the mode of pollination in a given crop and the extent of natural outcrossing, prediction of performance of double cross hybrids. This completes the second course. Let us move to the third one, that is GPB 211 Principles of Seed Technology. It has credit hours of 3, that is 1 plus 2. Theory consists of seed and seed technology introduction, definition, importance, deterioration, causes of crop varieties and their control, maintenance of genetic purity during seed production, seed quality, definition, characters of good quality seeds, then different classes of seeds, Foundation and certified seed production of important cereals and pulses, fodder, vegetables, oil seeds, then seed spices, seed certification, phases of certification, procedure for seed certification, and field inspections, Seed Act and Seed Act enforcement, duty and powers of seed inspectors, offences and penalties. Along with that, Seeds Control Order is also discussed, that is Seed Control Order 1983. Varietal identification through grout test and electrophoresis, molecular and biochemical test. Detection of genetically modified crops, transgene contamination in non-GM crops, GM crops and organic seed production, seed drying, processing, steps, seed testing for quality assessment, seed treatment, its importance, method of application and seed packing, seed storage, general principles, stages, and factors affecting seed longevity during storage, measures for pest and disease control during storage, then seed marketing, structure and organization, sales generation activities, promotional media, then factors affecting seed marketing, role of World Trade Organization and OECD in seed marketing. Coming to the practical sessions, seed production in wheat including seed standards, similarly in rice, maize, sorghum, bajra, urd, moong, cowpea, pigeon pea, lentil, gram, field pea, soybean, rapeseed and mustard, groundnut and sesame. Then seed production in vegetable crops like potato, cauliflower, tomato, chili. Then in fenugreek, fennel, cumin, coriander. Seed sampling methods, physical purity test, germination test, viability test, seed and seedling vigor test, genetic purity test like grow-out test, then electrophoresis, seed certification procedure, field inspection and preparation of field inspection reports. This has given quite more credits, like 2 means the amount of time contributed for teaching this particular topic. Then visit to seed production farms, visit to seed testing laboratories, and visit to processing plants. Coming to the fourth course, that is GPB 222 Commercial Plant Breeding. This has credit of 1 plus 2, that is 3 total. The topics include types of crops and modes of crop plant reproduction, line development and maintenance, breeding in self and cross-pollinated crops for development of hybrids and seed production, genetic purity test for commercial hybrids, advances in hybrid seed production of maize, rice, sorghum, pearl millet, advances in hybrid seed production of castor, sunflower, cotton, pigeon pea, brassica, then quality seed production of vegetable crops under open and protected environment, alternative strategies for the development of line and cultivars, that is haploid inducers, tissue culture techniques, biotechnological tools, IPR issues in commercial plant breeding, dust testing and registration of varieties under PPVFR Act, then varietal testing release notification system in India, principles and techniques of seed production, types of seeds, then quality testing in self and cross-pollinated crops. Coming to the practical lectures, the first one is floral biology in self-pollinated crops species, subsequently in cross-pollinated, then selfing and crossing techniques, techniques of seed production in self and cross-pollinated crops using ABR and two-line system, techniques in hybrid seed production using male sterility in field crops, understanding the difficulties in hybrid seed production, tools and techniques for optimizing hybrid seed production, concepts of line, its multiplication and purification in hybrid seed production, role of pollinators in hybrid seed production, then hybrid seed production techniques in sorghum, pearl millet, maize, rice, rapeseed and mustard, sunflower, castor, pigeon pea, cotton, vegetable crops, Sampling and analytical procedures for purity testing and detection of spurious seeds, seed drying, seed storage structure in quality seed management, screening techniques during seed processing like grading and packaging, then visit to public and private seed production units. Coming to the fifth course, that is GPB 311, crop improvement for Kharif. The theory part includes crop improvement aspects in rice as mentioned in the syllabus like centers of origin, distribution of species, floral biology, breeding objectives and procedures, etc. That includes hybrid seed production. Here I'll discuss in general centers of origin, distribution of species, wild relatives in different cereals, pulses, then oilseeds, fiber, fodder, cash crops, vegetable and horticulture crops, genetic resources, its utilization, conservation of floral biology, then study of genetic of qualitative and quantitative characters, important concepts of breeding self-pollinated, cross-pollinated and vegetatively propagated crops, major breeding objectives and procedures including conventional and modern innovative approaches for developing hybrids and varieties for yield, adaptability, stability, abiotic and biotic stress tolerance with quality like physical, chemical, nutritional quality, seed production technology in self-pollinated, cross-pollinated and vegetatively propagated crops. In cereals, the focus is given on rice, maize, sorghum and bajra, pulses, urd, mung, cowpea, pigeon pea, mothbean. Then in oilseeds, soybean, sesame, groundnut, fiber crops, cotton, fodder, bajra, sorghum, maize. In vegetables, chili and tomato, cash crops, castor, hybrid seed production technology in maize, rice, sorghum, pearl millet, pigeon pea, etc. Then idiotype concept and climate resilient crop varieties for future are also discussed in this particular topic. Coming to the practicals, emasculation and hybridization techniques in different kharif crops like rice, maize, sorghum, pearl millet, pigeon pea, urd bean, mung bean, soybean, groundnut, sesame, castor, cotton, cowpea and pearl millet. Maintenance breeding of different kharif crops, handling germplasm and segregating populations by different methods like pedigree, bulk, single seed descent methods, study of field techniques for seed production and hybrid seed production in kharif, estimation of heterosis, inbreeding depression and heritability, layout of field experiments, study of quality characters, donor parents for different characters, then visit to seed production plots, visit to AICRP plots of different field crops. Coming to the next course, that is GPB 321, crop improvement for Rabi, that is crop improvement part 2. This is also given 1 plus 1 means total 2 credits. One is practical and one is theory. Coming to theory part, this course doesn't much differ from the first course, but the crops covered are Rabi crops. So let us see the syllabus. Centers of origin, distribution of species, wild relatives in different cereals, pulses, oilseeds, fodder crops and cash crops, vegetable and horticulture crops, plant genetic resources, its utilization and conservation, floral biology, study of genetics of qualitative and quantitative characters, important concepts of breeding, self-pollinated, cross-pollinated and vegetatively propagated crops, major breeding objectives and procedures including conventional and modern innovative approaches for development of hybrids and varieties for yield, adaptability, stability, abiotic and biotic stress tolerance, along with quality like physical, chemical, nutritional, then seed production techniques in self-pollinated, cross-pollinated and vegetatively propagated crops. Remember, this is for Rabi season. This includes cereals like wheat, oat, barley, pulses like chickpea, lentil, field pea, Oilseeds like rapeseed, mustard, and sunflower. Fodder like berseem, oats, lucerne. Cash crop, sugarcane. Seed spices and medicinal plants are taken here like cumin, coriander, fenugreek, fennel, isabgol. Vegetables and horticulture crops like potato. Then hybrid seed production technology of rabi crops, idiotype concept, climate resilient crop varieties for future with respect to the rabi season. The practicals covered here are emasculation and hybridization techniques in different crops like wheat, oat, barley, chickpea, lentil, field pea, rapeseed and mustard, sunflower, potato, berseem, sugarcane. Then handling of germplasm and segregating populations by different methods like pedigree, bulk, single seed descent method. Study of field techniques for seed production and hybrid seed production in rabi. Estimation of heterosis, inbreeding depression and heritability, layout of field experiments, study of quality characters, study of donor parents for different characters, then visit to seed production plots and visit to AICRP plots of different field crops. Coming to the last course and the sixth course, that is GPB 322 Micropropagation techniques. This has also given 3 credits, that is 1 plus 2. Actually it is 2 plus 1. Here the theoretical part includes meaning and concept of in vitro culture and micropropagation, historical milestones, advancements and future prospects of micropropagation, totipotency, dedifferentiation, tissue culture methodology, sterile techniques, synthetics and natural media components, growth regulators, environmental requirements, genetic control of regeneration, plant regeneration pathways like organogenesis and somatic embryogenesis. Micropropagation, its definition, methods, stages of micropropagation and its significance, auxiliary bud proliferation approaches like shoot tip and meristem culture, organogenesis, its purpose, methods, requirements, indirect and direct organogenesis, somatic embryogenesis, procedure and requirements for organogenesis, indirect and direct embryogenesis, difference between somatic and gametic embryogenesis, synthetic seed concepts, necessary procedure and requirements for production of synthetic seeds, agencies working on micropropagation. Then practical includes laboratory organization, sterilization techniques for explants, glassware, plasticwares, labwares and working platform. Then preparation of stocks and working solution, preparation and sterilization of growth regulators, preparation of working medium and experimentation on determining the optimum concentration of growth regulators, callus induction and regeneration of whole plants from different parts of the plant, direct regeneration into whole plant using bud, node and other tissues, induction of somatic embryo, experiments of synthetic seed production and testing, storability and germination efficiency. This is all about the complete plant breeding syllabus for undergraduate program as proposed by the 5th Dean's Committee. So in the next chapter, in the next video, I shall discuss the syllabus for master's and doctoral courses. Thanks for watching. Keep on supporting me. Thank you.